Hey, I'm Bailey. This is my extra credit video on DNA replication, transcription, and translation. First, we're going to talk about DNA replication, which is just DNA replicating itself into more DNA. So first, the process has to be initiated by an initiator protein that attaches to the origin of replication on the template strand of DNA. Then the helicase enzyme attaches and breaks down the hydrogen bonds in between the strands and unwinds the helix. Then the single-stranded binding protein shown here hold the helix open so that it's easier for it to be replicated. Then during elongation, replication occurs from 3' to 5' on the template or forms in 5' to 3'. Along the template strand that's 3' to 5', there's a continuous strand of replication, and they are joined by RNA polymerase that catalyzes the phosphodiester bonds between the fragments. On the 5' to 3' of the template strand, they are replicated in short fragments as the DNA is broken apart and forms this replication fork. So as it opens, another fragment will form, and these are called Okazawa fragments. And they are binded together by DNA ligase. A bases join to T bases, and G bases join to C bases, and vice versa. Now we're going to talk about transcription, which is DNA to mRNA. For the initiation of this process, first a sigma factor has to bind to the promoter region so that then RNA polymerase can bind and go down the strand and make the mRNA. So in the promoter region, there's a thing called either a strong or a weak promoter. If the promoter is considered strong, a lot of mRNA is made because it allows the process to happen faster. And if it's considered weak, then slower because it makes the process slower. So then in elongation, RNA polymerase just moves down the template strand and adds the complementary code for mRNA. It's a little different than DNA code because the A now attaches to the U base, which isn't in DNA. And then the T still matches with an A, and then G and C stay the same. Then, for termination of the process, there's two different kinds of sequences that can end it. If there's an A-rich area in the template strand, there's a rho factor that binds and terminates the process. If there is a C-rich area, then it forms a hairpin loop, which just stops. And if it's A-rich is considered extrinsic termination, and the hairpin loop is called intrinsic termination. So now lastly we're going to talk about translation, which is the translation of mRNA into proteins. To initiate this process, first the AUG codon has to be identified, which is also known as the start codon on the mRNA. So the process occurs inside of a ribosome. Here's the ribosome with the large subunit and the small subunit. It moves down, the ribosome will move down the mRNA from 5' to 3' here, and as it moves down the mRNA, tRNA with attached amino acids attached depending on the codon on the mRNA. And multiple codons can specify the same amino acid. So different codes on the mRNA may code for the same amino acid attached to a tRNA. So during elongation, the ribosome just moves down the mRNA and attaches, and as the ribosome moves down, the tRNAs will detach and a polypeptide chain will release from the ribosome, and the mRNA just continues to move down. To terminate the process, there has to be recognition of a nonsense codon. Synthesis stops by these release factors, and all of the components release, and we're left with an mRNA, tRNA fragments, and a polypeptide chain.